Thank you, everybody, for coming to my talk today. My name is Rachael Wiedmeier. I am a master's student at Texas Tech University, and today I'm going to be talking to you about the characterization of desert bighorn sheep microbiomes in Texas. Bighorn sheep have had multiple mortality events across North America, and many of these can be due to the pneumonia complex. This is a polymicrobial community of pathogenic bacteria. Most research has been done on Mycoplasma ovipneumonia, or I will refer to it as MOVI for the rest of this talk. But because it is polymicrobial, we have a few other bacteria that are a part of this disease. So Manihemium hemolytica, Pasteurella multocida, Biebersteinia trehalosii, and Fusobacterium necrophorum, along with Fusobacterium subspecies, are all part of this disease. All of these, besides MOVI, are found in healthy bighorn sheep in their upper and lower respiratory tracts. So something about MOVI is thought to be a predecessor for the rest of these and somehow changes the environment and makes the rest of these opportunistic pathogens. So this complex is transferred usually through social interactions. This is any kind of muzzle-to-muzzle touching, swapping of saliva and or mucus, and this can become airborne for short distances, but for the most part, it's usually just any kind of muzzle-to-muzzle interaction. The biggest management implication is domestic sheep and domestic goats that carry all of these pathogens asymptomatically. Another management implication has been adult bighorn sheep that survive mortality events and become carriers of these pathogens. So most of the time, this is just used to their lambs. So if a ewe survives the mortality event, moves to a different herd, and then ends up giving birth to another lamb because they spend so much time together, that's usually how that interaction and transfer happens. But it has been known to be transferred through rams who move to different mountain ranges after they leave. So that brings us down to the American Southwest. Why do we care down here? Well, of course, many of you know we have aoudad, which is an invasive species to Texas. Aoudad are in the same subfamily of Capernae as domestic sheep and domestic goats and bighorn sheep. And so due to that closely relatedness to bighorn sheep, this makes them a possible transmitter of disease. So for my study specifically, I hope to identify a baseline of microbiome health for desert bighorn sheep and aoudad here in Texas. So that way, once we know what a healthy state looks like, we can quickly identify what a disease state looks like. I hope to be able to create a sampling and data processing method that's easily replicated in the field. I hope to report on any pathogens that are novel in our desert bighorn sheep here. I want to identify any spatial patterns among our microbiomes, and then I'd also like to evaluate how the presence and absence of MOVI correlates with other pneumonia complex members. So again, MOVI is thought to be a predecessor, so how does that change the environment, and how does that change the rest of our pneumonia complex members, like their relative abundances, to make it more opportunistic pathogenetic? So for my data collection, we opportunistically sampled bighorn sheep and aoudad using helicopter captures between 2016 and 2020. And on these captures, we collected throat and nasal swabs. So my sampling distribution includes 10 different mountain ranges. This is the Baylor Mountains, Sierra Diablo, Beech, Van Horn, Sierra Vieja, Bassi, Chinades, and Dove, and then Elephant Mountain WMA and Black Gap WMA. This is a total of 366 individual animals, which breaks down to 278 bighorn sheep and 88 aoudad. For all of our individuals, we are able to get a nasal swab, but due to how we wanted to swab the throats, we were only able to get 109 throat swabs from bighorn and 41 from aoudad. After we collected all the swabs, we sent them off for 16S sequencing to identify bacterial species. This is just a specific loci for bacteria. Once I got all those raw sequences back, I filtered and sorted them down to a strain level, and then I assigned taxonomies to each of them. So the first thing I wanted to do was look at what are our most common bacterial species. So for our nasal swabs here, these are the top 10 most common bacterial species in all of our nasal swabs. Ironically enough, all of these are actually bacteria that are isolated in soil. So I've never personally been net gunned, but I can imagine when you do get net gunned, you probably get your face pretty close to the ground. You might get a little bit of dirt in your nose. So what we probably had was just some dirt on our swabs. No big deal. What we can do is actually create a database downstream of all soil bacteria and actually filter them out. And I can still get relevantly biological data that we'd expect to see in a bighorn nasal cavity. So moving on to our throat swabs, we do see something a little bit different here. So all of these on the screen are considered opportunistic pathogens. Notice that we do have Biebersteinia trachelosi, Fusobacterium species, and Manihemi hemolytica. So those are all part of the pneumonia complex, and all 6 of these are actually known to cause either endocarditis or any kind of infection in GI tracts or respiratory tracts. So again, we are seeing some pathogenic bacteria. So the next thing I wanted to do was look at what are the top 10 most common shared bacteria between both audad and bighorn sheep. So in our nasal swabs, again, we did see a lot of soil bacteria. No big deal. We'll filter that out downstream. We did see one G. bovis, which is isolated from cattle with digital dermatitis lesions. So this can be a pathogen, possibly. So moving on to our throat swabs, we do see a few soil bacteria in here, which is expected. We're capturing these animals in the same locales, and because your nasal cavity is attached to your throat, you're probably going to have eventually some soil move down there. So we are expecting to see that, but I can filter that out downstream. But we do still see a few pneumonia complex members in here. So again, some Fusobacterium, Manihemi hemolytica, and Biebersteinia trachelosi. So this is a nasal microbiome alpha diversity plot. Down here, I broke it down to our 10 different mountain ranges here on the y-axis, and then our observed number of bacterial species on the x-axis. The biggest thing I want to note here is the Chinades and the Sierra viejas. So you'll notice that these are much lower in observed number of species, and this could just be due to how the samples were taken. These specific locations were actually taken by the capture crew in the field. So anybody who was in the helicopter was able to take those samples for us. So it was just a little bit of differing in sampling technique. And then our largest sampling size is going to be our Elephant Mountain WMA. So this is a PCOA of our nasal microbiomes by capture year. This is just bighorn sheep. So the only locale that we were able to get multiple years sampled was Elephant Mountain WMA. The rest of these, unfortunately, due to opportunistic sampling, were different years. And I'd like to note that we do have a few that have smaller sampling sizes. Just due to, again, opportunistic sampling, we were able to get what we could get. So I ran a overall permanova for mountain range and sex, where we found sex insignificant, but then we did find the mountain ranges to be significant. So from there, I broke them down to their pairwise comparison testing on the mountain ranges, and I adjusted the p-value for multiple testing corrections. These were the ones that we did find significant, so we're only going to focus on those. So again, going back to our PCOA of our bighorn sheep nasal swabs, we can see that Van Horn clusters in a very different way, and they were completely different than almost all of our other localities. We also saw our WMAs to be different, which is good to note due to how Texas Parks and Wildlife actually manages the sheep. So they often take Elephant Mountain sheep and will take them to Black Gap. So it's good to know what our starting point is and where they're going to be going. And then we also found the Diablos to be different from both of our WMAs. So this plot here is the relative abundances of pneumonia complex members based off of if that individual is carrying Mycoplasma ovipneumonia or not. So we did have 2 audad that were identified to be carrying Mycoplasma ovipneumonia. That will be these 2 red dots that are hovering over the true section in each of our 4 plots. And then the rest of these hovering over the false are all individuals that are not carrying MOVI. So the biggest thing I'd like to note here is our Biebersteinia trachelosi and our Fusobacterium necrophorum. Those audad that did have MOVI are at a 0 relative abundance for those 2 specific bacteria. And the rest of these actually have a little bit higher of relative abundances. So especially in our throat swabs. So that is really good to note that maybe these are very healthy bacteria, and when they're gone, that's a bad thing. Our Pasteurella multocida is actually quite high for the 2 audad that we're carrying MOVI. We also have these 4 that are audad, and as of right now in my data, they are still alive. I am waiting on some collar data to come back, but as far as I know, they are still alive. The last thing I'd like to talk about today is our Mycoplasma ovonemona phylogenetic tree. So all of these sequences here in blue and green were actually sequences that were published in a paper from Kamath in 2019. These are all MOVI sequences that have been found in either domestic sheep, domestic goats, or bighorn sheep, Rocky Mountain bighorn sheep. These in blue are considered from domestic sheep, and these in green are considered domestic goat strains. Ours are in red right here. So again, we had the two audad, but between those two audad, they both had carried 26 different strains of MOVI. So that just means there are 26 different bacterial DNA sequences. Ours lied most closely related to the domestic goat strain, but even from there, we had our divergence at 2.8%. So what this means is we have almost a brand new bacterial species down here in our audad. At a 3%, that's usually when bacteria species are identified as a completely different bacterial species. So what this means is we could either have a more pathogenic strain, maybe a more pathogenic or less pathogenic strain to our sheep or audad. Really, we don't know. It's brand new. So for our preliminary conclusions, we found that a lot of our nasal swabs were carrying a lot of soil bacteria. No big deal. We can rectify that by creating a database full of soil bacteria and filtering them out downstream. And then we do have two audad that were carrying MOVI. It is possible for audad to carry MOVI. The strains that we have are very different from Rocky Mountain bighorn sheep strains, and that could make them possibly more pathogenic strains than those that have been encountered before. And with that, I'd like to acknowledge the lives of Dr. Dittmar, Dewey Stockbridge, and Brandon White. They were amazing coworkers and friends. I didn't spend a whole lot of time, but the time I did get to spend with them in the field was always full of laughter and smiles and fun. And Dr. Dittmar was a great mentor to me. I would not be anywhere near where I'm at now without his help and his great advice. So they are greatly missed. We'd also like to thank Texas Parks and Wildlife, BRI, Texas A&M, the NSRL at TTU, the Texas Bighorn Society, and the Wild Sheep Foundation. Help from Phillips Lab, Bradley Lab, and Conway Coalition, and our funding sources of the Texas Bighorn Society, the Wild Sheep Foundation, the Rumsey Research and Development Fund, the Bricker Endowment for Wildlife Management, and the Department of Biological Sciences. And with that, I will take any questions. Thank you for coming to my talk today.